So hello, everyone. It's really a great pleasure and honor to start 2026 Immune Talks. And with us, we have a very special guest, LiFan Lu, which I'm going to introduce in a moment. So thank you, LiFan, for joining us and for sharing your passion and research. Um, I just like to remember that this year we have a very special way of actually having live question at the end, which we didn't have before. You can still ask your question that will be accompanied with the YouTube video, but there is also an option when the meeting ends to have to ask a question on live, and I will share some of my questions as well. So looking forward for the question, and Lifan is also looking forward for the interaction with you. So please mark your name and ask the most challenging question to Lifan at the end. So be ready, Lifan Lu, for this exciting tour. So just for a short introduction, and you can add anything that I missed. So Lifan Lu has his own research group at UCSD. And in his research, he explored the molecular and cellular mechanism underlying immunological tolerance to self-antigen. And this is quite an exciting year to Tregs and their biology following the Nobel Prize. So I guess these cells do mean something, apparently. And and of course, he has many very important publication on these subject and really introducing many critical mechanism biology on Tregs and their role. So without further ado, Lifan, the stage is yours.
Um, hi everybody. Thank you for giving me this opportunity. And I just realized Ido and I wear very similar clothes, and we didn't plan for that. But yeah. So should I start, or Ido, are you going to ask a question first?
Oh yeah, I forgot. I should also send you a shirt with our lab logo. We'll do it next time.
Yeah, I mean we have the
Yes.
same style. I mean we really didn't plan for this.
Yeah, it wasn't planned. Absolutely. I guess we were synced before the meeting. So just one question. Yeah, I wanted to ask you. Can you share sort of, I think, for the younger crowd, the moment when a failure or a negative result or any or something that you didn't expect, was not uh, was not turning you to a bad direction, but really changing the way you thought about the science and the discovery, and you sort of directed it to a positive way, to a
Yeah. Thank you. So yeah, I think first of all, I think I want to clarify. I feel it's not fair to put value and negative result together, right? So I think when we do research, the value means inconsistent results. Negative result is actually not bad. Actually, negative result allow you to move on to somewhere else. Inconsistent result to me is much worse than negative results. So, but I do have a short story, not short story, answer for this question. So actually, when I did my PhD work with Dr. Randy Noel at Dartmouth, initially I was I was planning to study CD40 signaling. So CD40 signaling obviously is very important, and Randy uh, is an expert in CD40. Uh, and so at the time when I joined the lab, the project that Gabe was kind of introduced to me was to identify the novel downstream uh, molecule downstream of CD40 signaling, because there was a publication just published from the lab showing that the known trap molecule, trap2, 3, 6, the binding site after they get disrupted in CD40 intracellular domain, CD40 still have some residual function. So Randy told me, oh, there must be some novel downstream signaling molecule that CD40 can signal through. So that's my first project. And then I did everything I could at the time, and then in the end, still finding is trap. It's just binding through a novel binding domain. So the known binding domain get disrupted, but they can still bind to a novel binding domain under in the CD46 intracellular domain. So it's not that exciting, right? Because it's still trapped, it's not a novel molecule. So you're a little bit disappointed. But then that's just the end of my second, third years, and then I still have a few more years to go. So what should I do? At this point, it's kind of a dead end. But then Shimon Sakaguchi published a paper showing GITR is important in Tregs. And GITR is another TNFR superfamily members. So I went to Randy and said, well, I have spent all my time in study CD40 signaling. Now GITR seems to be very important in regulatory T cells, and it's another TNFR superfamily. Perhaps I can use my knowledge in CD40 signaling and apply to the study on GITR in Tregs. And that's how I switched to study Treg. And of course, the rest is history. But and today the talk actually I'm going to give is nothing about Treg. It's also the byproduct out of our initial study. You want to study Treg. So I guess that's the way I look at negative result. Negative result is a positive result in somewhere else. It's just not the place you were looking for in the beginning. So that's my answer.
Exciting. Thank you. Thank you for sharing this.
So I will share my slide. Okay, hold on a second. Can you see my slide?
Yes. Great.
Okay. I think I would just get started. Yeah. Okay. So again, thanks for the organizers and everyone involved in Global IMMUNOTALKs. It's really my pleasure to share our work, mostly unpublished today. The title, as you can see, is Talks in the Immune System. As I mentioned a couple of minutes ago, even though I spent a lot of time working on regulatory T cells, but today is actually not about regulatory T cells. It's more about TOX and TOX family members in immune regulation or immune cells outside regulatory T cells. So, okay, so let's see. So, so within the TOX family, there are 4 family members. Most study members are TOX and TOX2. Well, TOX3 and TOX4 are obviously under study at the moment. TOX3, especially in the immune system, the role of TOX3 in the immune system hasn't been really studied. It's mostly for their, so far what we know about TOX3 is for their role in either breast cancer or in the neuronal development. And TOX4 has been shown to be involved in chromatin structure, remodeling, but what we know about TOX family members today in the immune system is mostly about TOX and TOX2. And in very early 2000, and the work done by Jonathan Kyle at Cedars-Sinai have clearly shown a critical role of TOX in the thymus, particularly for their function in CD4 lineage T cell development. So what he has shown that in the absence of TOX, that you do not get CD4 T cells and NK T cells and Tregs, but you will have still CD8 T cells, even though they're still somewhat negatively impacted, but they're there. And in the periphery, obviously, you can still find CD8 T cells, but you couldn't find CD4 T cells. And then later on, they also continue to show TOX is not only important for CD4 T cell lineage differentiation, and also show TOX is important for NK cells and innate lymphoid cells. So in the absence of TOX, you have this drastic defect in all innate lymphoid cell subset NK cells differentiation. So you lose all these populations. So this result suggests TOX is really serve as an important molecule upstream of many different immune cell lineages. Of course, what we know about TOX today is mostly based on a series of findings back in 2019. at the time, the role of TOX in peripheral CD8 T cells rather than in the early CD8 T cell development in the thymus, it was demonstrated. So basically, what was shown in those studies is TOX is induced upon continued T cell stimulation, leading to NFAT activation. And that TOX induction would drive this what we call CD8 T cell exhaustion. So there are many studies following those initial studies back in 2019 demonstrating TOX is that one, if not the most important, but at least one of the most important transcription factors in the establishment of CD8 T cell exhaustion, either during chronic infection or in cancer. But of course, as I said, without TOX, the early study has shown that you don't get CD4 T cells. That's why there's not much known in terms of the role of TOX in CD4 T cells, since everybody focuses on its role in CD8 T cells. So in our lab, we have found in CD4 T cells, if you look at this specific subset of CD4 T cell called follicular helper T cells, especially the one in the germinal center, the germinal center TFH, we can see very high level of TOX expression, both in human and in mice. So basically, this suggests that TOX can also have a role in peripheral CD4 T cells. So to test that, in that study, what we did, obviously you cannot knock out TOX in the very beginning, so we either use the CRISPR-Cas9 to knock out TOX in the peripheral CD4 or use the retrovirus to overexpress TOX in the peripheral CD4 T cells. And we can see TOX is functionally required for TFH cell differentiation. So loss of TOX give you fewer TFH, you give more TOX, you get more TFH cells. As I said, this study was not initially intended to study TOX in TFH cells. It actually is a byproduct of our other study, try to study microRNA in T follicular helper cells. But in any case, we found in this study, TOX is induced by BCL6, and then it can drive the expression of many downstream molecules like TSF1, LEF1, PDCD1, which are all contribute to TFH cell differentiation. So at the same time, there's another group like Chen Dong have shown independently another family member, TOX2, is also required for TFH cell differentiation. And basically what they show, similar to what we found in TOX, is that BCL6 is also driven by BCL6, just like what we show in our TOX story. And then TOX2 can also lead to the establishment of TFH cells differentiation program. And they also show that in the same study that both TOX and TOX2 have this kind of a synergistic effect, that deletion of one lead to reduced TFH response, but deletion of both lead to either even further reduction of T follicular helper cell response. So basically suggests that both TOX and TOX2 contribute, and they actually work synergistically to promote TFH cells differentiation and function. And this synergistic effect between TOX and TOX2 is not only found in TFH cells, but is also found in CD8 T cell exhaustion. So this is a study that done by our collaborator and Jenna Rao at LJI have shown that if you delete TOX and TOX2 together, you are able to make exhausted CD8 T cell functional, more functional than deletion only one of them. And essentially, in this study, they show both molecules also contribute, and there's a synergistic effect between these two molecules. So far, all these studies done by many groups, including us, kind of pointed to TOX as a key regulator for regulating immune cells development and function in both central lymphoid organs and peripheral tissues. On the other hand, though, TOX2 appear to be only impact on the immune cell in the periphery, because deletion of TOX2 doesn't affect CD4 T cell development in the thymus, doesn't affect innate lymphoid cell development in the in the bone marrow. So basically, it seems TOX and TOX2 have similar role, but at the same time distinct function, where TOX affect not only peripheral but also immune cell in the central lymphoid organ, while TOX2 is mainly acting on peripheral immune cells. And this finding is not only can be found in the adaptive immune system, it's also been shown recently in the innate immune cells, particularly innate lymphoid cells, type 3 innate lymphoid cells. So there's a recent study showing that unlike TOX, sorry, the citing is wrong, it's not 224, it should be 225, is unlike TOX, TOX2 is specifically required for only a subset of ILC3. So the subset of ILC3 that reside in the gut. Okay, so without TOX, as I said in the very beginning, that you have a broad effect in all innate lymphoid cells. But deletion of TOX2 affect only a subset of ILC3 in the intestine, not even affecting ILC3 outside the intestine. And this was shown it was shown in this paper is driven by HIF1 alpha, and that is important for the gut innate lymphoid cell type 3. And in that study, what they found is reduced ILC3 responses in the toxic deficient mice lead to impaired host defense against Citrobacter infection, right? So you can see this reduction of ILC3 in the gut, and that have a the poor immune response against this enterobacterial infection. So now, the type 3 immunity, as we know, is composed of not just ILC3, another immune cell population in the adaptive immune population, Th17 cells, are also known to play a very important protective role during Citrobacter infection. So our question at the time was, could TOX2 deficiency also lead to dysfunctional Th17 cell responses? And that also contribute to the impaired host immunity in the TOX2 deficient mice. So when we look at the expression level of TOX2 in different T helper cell lineages, and as we know, I showed you in the previous slide, TOX2 has been shown to be highly expressed in TFH cells by Chen. And even though in vitro you couldn't really differentiate TFH cells, but you can get something close to TFH cells, which you call TFH cells-like cells, by giving IL-21, sometimes combined with cytokine IL-6, and that can give you some level of TOX2 induction, right? But if you really look at this, the cell population that have the highest TOX2 expression is actually Th17 cells, much higher than other TH cell subsets. So the question is obviously whether this is just a mouse phenomenon. The next question is, can we also see that in human? Can we also see that in vivo? And then so to do not just the in vitro, we also look at the in vivo model, which we employ this anti-CD3 induced intestinal inflammation model, where it has been shown by many groups, including Richard Ravel back in 2011, that if you give mice 3 shots of anti-CD3 antibodies IP, you're able to induce a Th17-driven small intestine inflammation. So by using this method, we are able to show that if you gated on the CCR6 positive IL-17 enriched population, you are able to detect high level of TOX2 along with our gamma T. Where in the CCR6 negative population, which you don't have Th17 cells, you don't see that much of an induction. And this induction is not just due to anti-CD3 injection, because we know if you give anti-CD3 to germ-free animals, in that situation, you don't drive a Th17 response, this TOX2 induction was abolished. So even you can gate on CCR6 positive, you don't see TOX2 induction. So now we show the expression, but is it functional, right? So first, we use the gain of function approach. We overexpress TOX2 in the T cells, and then polarizing them to TS17 cells. As you can see, if you overexpress TOX2, you are able to get more TS17 cell out of the TS17 polarization condition. However, TOX2 overexpression itself is not sufficient to drive TS17 differentiation, as you can see in this group where cells are cultured under TH0 condition. Doesn't matter you have TOX2 overexpression or not, you don't get TS17 cells. But it can enhance or promote more TS17 cells if the cells are under TS17 polarizing condition. So this is gain of function. And how about loss of function? So we create a T cell conditional knockout mice where TOX2 is deleted in old T cells. And as you can see in vitro, we can see deletion of TOX2 will lead to the reduction of TS17 differentiation. And that is not only can be found in vitro, but it also can be found in vivo, which I will show you later, sorry. But first, I want to say that this, we believe, is a defect of differentiation rather than a defect in their proliferation and survival, because when we look at those TS17 cells, they have the same frequency of Ki67 positive population. They also do not show increased death. So we believe that TOX2 is involved for TS17 differentiation rather than affecting their survival or proliferation. So as I told you that was done in vitro, what happened in vivo? So again, we went back to this NACD3 mediate small intestine inflammation model, and then we give those wild type and knockout mice with NACD3. As you can see, those mice, they don't have the same level of pathology that you normally found in the wild type mice, and they also did not lose weight as fast. And look at the TS17 cells within the small intestine of those TOX2 T cell conditional knockout mice. They also have reduced TS17 cells, as shown here as IL-17A, and we also found IL-17F and other TS17 associated cytokine production, as well as, of course, the transcription factor R gamma T. But in terms of other cytokine like interferon gamma, we really don't see a clear difference. And that's not just in the intestine. So unlike what was shown in ILC3, in TS17 cell, the effect of TOX2 seems to be much broader, because you can also look at TS17 cell in other tissue, like in the brain. When we employ the EAE models, where we can show that the TOX2 T cell conditional knockout mice also have attenuated disease, as shown in the weight loss and the disease score. They all have attenuated disease phenotype compared to the wild type. And same thing, if you look at the cytokine response, they have reduced IL-17A production, reduced GM-CSF production. And again, we do not see a difference in their interferon gamma responses. So the data so far, we suggest that TOX2 is required for optimal TS17 cell responses. Of course, the question is how, right? What is the mechanism? And how does TOX2 control TS17 cells? So to address this question, because lack of proper tool so far, so we went ahead to generate another mouse model where we knock in both the TY tag in the C terminal of TOX2 as well as a GFP. So this mouse allows us to first can track TOX2 expressing cells in vivo. As you can see, that indeed IL-17A positive cells are TOX2 GFP positive, while the IL-17 negative cells are GFP negative. And then we can also use the antibody, anti-TYY antibody, to pull down endogenous TOX2 for any biochemical study we want to do. And if you look at the GFP expression as a surrogate for TOX2 expression, and then also R gamma T expression side by side at different time points during TS17 differentiation, you can see that both TOX2 and R gamma T are highly induced around the 24 hours after you put that under the TS17 polarization condition. And deletion of TOX2 can already see some effect at 24 hours, even though it's not as clear-cut, statistically speaking, but it's clearly needed, as you can see, follow 24 hours, 48 hours, and 96 hours, there's a much reduced R gamma T expression in TOX2 deficient T cells. So at least from this, we know TOX2 is needed to promote and sustain R gamma T expression during TS17 cell differentiation. But is it direct or is it indirect? So having the mouse model I just talked about, we are able to perform the cut and run study to identify the right TOX2 target in the TS17 cells, as well as combined with H3K4 trimethylation cut and run, then we hope to identify direct TOX2 target. As you can see here, the Venn diagram analysis show there are 5,202 unique peaks that the common peaks that are shared by the TOX2 binding and the H3K4 trimethylation. And then within that 5,202, there are 2,466 unique genes, including RRC. And we can confirm that by doing luciferase reporter assay that the binding is functional. So TOX2 indeed can directly bind to RRC and then drive its expression, but not just RRC, but many other known TS17 associated genes like RA, like TSF3, RAIL-A, RBPJ, are all be found to be the TOX2 direct target, and those bindings are also functional. So this, again, at least R gamma T, among others, can be directly targeted by TOX2 in TS17 cells, and that lead to TS17 cell differentiation. But of course, we want to validate it. So can enforce expression of gamma T back to the TOX2 deficient T cell rescue the defect that we observe. So we perform the experiment, again, by retrovirulously overexpress RRC back to the TOX2 knockout, shown on the left side. You can see now we can eliminate the difference between the wild type and the TOX2 deficient T cells. They are all expressed very high level. However, if you look at the IL-17 response, while overexpression of R-GART-T in TOX2 deficient T cells can lead to increased IL-17A production, but it's still lower than the wild type T cell overexpressing R-GART-T. So that suggests that even when we overexpress R-gamma T to the level similar to wild type with R-gamma T overexpression, they still have a defect, despite R-gamma T can partially rescue the IL-17 production. And this is actually supported by our observation. Simply look at TOX2 T cell conditional knockout mice, gated on R-gamma T positive cells, which we know they have reduced R-gamma T positive cells. But if you only gate on R-gamma T positive cells, you can see that even within R-gamma T positive cells, they still produce less IL-17A, as well as many other IL-17 associated cytokines. So that suggests that R-gamma T expression alone is not sufficient to explain the phenotype we observe in TOX2 deficient T cells. So something else, in addition to R-gamma T, TOX2 regulate is important for the optimal TS17 effector function. So to address this, then we went back to perform the RNA-seq and try to get a kind of a picture of TOX2 dependent genes. They may or may not be a direct target of TOX2, but nonetheless is dependent on TOX2. And first of all, you can see RRC certainly is downregulated in TOX2 deficient TS17 cells. And then IL-17A and IL-17F, which are not the direct target of TOX2, are also downregulated in TOX2 deficient T17 cells, which is consistent with what we saw previously. But then when we, again, perform Venn diagram analysis to combine the TOX2 direct target and the TOX2 dependent genes, as well as the Th17 gene that has been shown in the past, and then we can basically separate those genes into two groups. One is a direct target, including RRC. The other one is not a direct target, but TOX2 dependent, which also associated with Th17, including IL-17A, IL-17F, and also MALT1. And the reason why we're interested in MALT1, because MALT1 has been previously shown to be required for Th17 effector function, but not their differentiation. So MALT1 deficiency doesn't affect R gamma T induction, doesn't affect their other Th17 gene program, but is required for their cytokine production. So we can confirm MALT1 by qPCR is indeed downregulated in the TOX2 deficient Th17 cells. And as I just told you, it was shown that MALT1, which is an important protease and structure protein upstream of NF-κB activation pathway that is needed for Th17 effector cell function. And the other downstream genes that we can also see are also dependent on TOX2, like JCAM and NF-κB. So again, we went back to our rescue study. Now, instead of overexpressing R gamma T, we overexpress MALT1. Again, overexpression of MALT1 in both wild type and the knockout can eliminate the difference between them that's shown in the empty vector group. And now with the same level of MALT1 expression, the TOX2 deficient T cells now are capable of producing the same amount of IL-17A as well as other IL-17 associated cytokines in the TOX2 deficient T cells. So now we can say that there are at least two molecules downstream of TOX2 are required for proper Th17 biology, including R gamma T, which is a direct target involving Th17 differentiation and function, but also MALT1, which is not really required for R gamma T induction or Th17 differentiation, but play a key role for optimal Th17 effector function, which, despite not a direct target of TOX2, is dependent on TOX2 expression. Now, we go back to our initial question about TOX2 and TOX in general, is we know that they are important in many other cell types, including T follicular helper cell and CD8 T cell exhaustion, in terms of TOX and TOX2, as I said in the very beginning. So far we show TOX2 is required for Th17, but does TOX also play a role in Th17 cell biology? And it does. So when we perform the gain of function study, compared with TOX2, it's not as good as TOX2, but overexpression of TOX can also promote Th17 differentiation. As you can see, increased IL-17A and increased R gamma T. But it's not as good as TOX2. So then you wonder, maybe it's just it have role, but not as important. But this is gain of function. So to really assess the requirement of this gene in a particular cell type, of course, loss of function study is necessary. So to that end, because you cannot delete TOX in the very beginning, so we generate the CD4 ERT to create TOX flux mice. In this mouse model, we can delete TOX in peripheral T cells by giving tamoxifen. Then with this treatment, now, again, you can see a very strong defect in Th17 responses as shown in IL-17 production as well as RRC expression. So then both TOX and TOX are important, but do they also function synergistically in Th17 cells like what we other people have shown in TFH and exhausted CD8. So to address this question, we delete both of them, just like other people did in CD8 T cell study and TFH study. However, unlike what they have shown in CD8 and TF helper cells, in TH17 cells, they do not seem to have additional effect when you delete both. Essentially, deletion of both molecules, the T double knockout mice, give you the same defect in TH17 cells when you delete only one molecule. Either deletion of TOX or deletion of TOX2 have the same effect, both in vitro and in vivo. So how to explain that? What we found is actually TOX and TOX2 can directly interact with each other in TH17 cells. So in human and in mice, you can use TOX2 to pull down TOX, as shown by co-IP study here, that when you IP use the anti-flag antibody, you're able to pull down TOX2 and then TOX will go with it. So that suggests that TOX and TOX2 can directly interact. But this is obviously in vitro overexpression. So how about endogenous TOX2? So we perform the IP mass spec to pull down the TY1 tag TOX2 and then perform this mass spec study. And as you can see here, the top molecule that can be pulled down together with TOX2 is TOX. And another molecule you can see is interesting, is TOX4, another molecule in this family also is one of the top targets. So it seems that the reason we don't see additional effect is because TOX and TOX2, they interact together and form a complex. And this phenomenon is not only being shown by our study, it actually has been shown by an independent study in other cell types back in 2021. When they use TOX2 as a bait, they are, I mean, in this case, it actually is the HEC293 cells. And they can pull down many molecules that would also shown in our mass spec study, including TOX4 and several other genes that you can also found in our own mass spec study in T cells. So altogether, we believe how it works is TOX and TOX2 forming a multi-protein complex, and together they can directly target genes like R-gamma T and many other TH17 associated genes to influence their differentiation. But that's not enough. They also regulate MOF1, which is required for optimal TH17 effector function. So to summarize what I just told you, which I show you TOX2 is highly expressed in TH17 cells, both in vitro and in vivo, and overexpress it lead to enhanced TH17 responses with deletion of TOX2 resulting in paired TH17 responses. And in the absence of TOX2 in T cells, you have attenuated TH17-driven diseases, not only in intestine, but also in the brain and other tissues. And we believe it facilitates optimal TH17 differentiation and effector function by directly targeting R-gamma T and other TH17 associated molecules like MOF1. And in the end, I hope I convinced you that unlike what people found in TFH cells and exhausted CD8 T cells, we believe that in TH17 cells, at least, TOX and TOX2, they form a multi-protein complex and together control TH17 responses. So in the end, I want to thank the people who were involved in this work, including my postdoc, Nimi Marcel, shown in the picture, who is the main driver of this project, and two other postdocs, Chen-Zhang Wu and Wei-Zhen Yan, who started this project, and also our collaborator at UCSD, Amgen, Taiwan University, University of Utah, and in Munich, who all contribute to this project. Of course, my funding, and I will be very happy to take questions.
This is beautiful, beautiful talk. Thank you so much for sharing this new work. I haven't seen it published yet. It's not.
No, it's unpublished. It's unpublished.
Yeah, beautiful, really, and some unexpected results. So, um, we can, I mean, the question can be asked live. There are around 200 participants. So if you wanna, some of them have posted their questions. So if you wanna ask the question directly, I think that's the best way. And you know, like old times we used to actually meet more often. So if anybody wants to ask directly the question, go live and ask. That's a wonderful opportunity now.
Um, should I just answer based on the order, or you will pick the questions?
Ah, so it's not possible to directly answer. So I will let's do I ask one and then you ask yourself one, so it'll be a bit more interesting. So
So
Daniel Kim
yeah.
asked, okay, we'll start with this question. Is TOX2 not involved in thymocyte development, similar to what we have with stocks? And would this in the Cree example change the number of CD4 cells?
No, as I said, like it's not just CD4 T cell, but also many other immune cell type where TOX has been shown to be important.
Mhm.
TOX2 deletion doesn't really affect CD4 T cells development in the thymus. We couldn't see any differences. And that's why in my early slides I mentioned there's a distinct difference between TOX and TOX2, where TOX seem to be important both at the early phase as well as later phase during in the periphery. Well, TOX2 seem to be more specialized in peripheral immune cells. And that's what we found.
Got it. Beautiful. You want to ask the next question by AC? There's no full name, so we just go by the AC initials.
Okay, so basically the question is about the R gamma T positive gamma delta T cell. In is T17 expression rescue? I'm a little bit confused about the question. Um, um, yeah, I actually I do you want to ask whether there's also a defect in our gamma T positive gamma delta T cells or I don't know
I think
about
that's how I understood the question. That's why I
yeah
pointed to you
because you talk about TFH cells, I don't
yeah,
understand what.
I don't
Um,
think yeah, go ahead.
Yeah, so we haven't looked into enough deeply to really to say the differentiation of function, but generally speaking, the R gamma T at least on the T cell population, we can see a similar effect, but it's not the same degree, and we like I said we haven't looked close enough, so I don't want to to make a big statement of it. But I think in general, I think TOX2 is involved in type 3 immunity.
Mhm. Got it. Another question by Zai Li. Um, and they're asking, he or she's asking, is TOX AKT3 interaction functionally relevant?
Um, um, I don't have the answer for it. And obviously, as you can see, our story is more about TOX2, at least in TS story,
Yeah.
even though we can see TOX have the same effect when you delete them. But and we believe they work in the same complex. But in terms of TOX and AKT3, I don't have enough knowledge to answer the question. Sorry.
Okay. Thank you. Do you want to take the
Yeah,
TH17
so
question?
our TSM generally come from specific tissue. And yeah, so that's what I said. We look at in the intestine, both small intestine and large intestine. We look at in the brain and the spinal cord in the EAE model. We see the similar TOX2 induction in TSM cells. And you can also see in the spleen. Of course, you can get very much fewer cells in the spleen. But basically, we can see that TSM cells have higher level of TOX2 regardless where they come from. And the part I didn't talk about it today is like we also look at pathogenic versus non-pathogenic TSM cells because this is one of the topic in recent years, and they all both show high level of TOX2 and TOX actually. So it's not just TOX2. I didn't talk about the TOX expression level, but they have the similar induction in TSM cell again, regardless whether where they come from or whether they are pathogenic versus non-pathogenic.
Cool. So I have another question by Shubhada Chiplunkar. I hope I'm pronouncing it correctly. What happens to TOX2 in Tregs, considering TH17 Tregs are
Yes,
inversely related? Yeah.
that's a very interesting question, which obviously we are also closely following. So we have another different project, right? So try to look at TOX and TOX2 in Treg, particularly you can argue that in the R gamma T positive Treg, also in T follicular regulatory cells, considering the role of TOX and TOX2 in TFH. So and based on what we have found, again, it's still very early. They are defect in TFR and T R gamma T positive Treg. And their function to control TS17 cells. So it's very interesting. Again, it's like many other Treg studies, that when you delete a gene in Treg, where the gene has been shown in other T helper cells type, there's always a phenotype that you can see, and that's what we saw so far. But again, it's very early in terms of the story we have so far. That's why I will wait more until we collect more result to be more certain.
Yeah, great. You want to take the question by John Hendry?
So can TOX2 interact with GATA3? We didn't see GATA3 in our mass spec data. So and if you look at the other mass spec study, as I published back in 2021, the Cell paper, I don't think GATA3 was identified in that complex in another independent group studies. So I don't know. Again, mass spec, you can always argue, even though you don't see them, doesn't mean they cannot be there. So I don't know. I mean, of course, we can do the overexpression study, but again, you can argue whether the overexpression study is artificial. Even you can see the interaction, whether it really happened at the endogenous level. This always can be debatable. But at least I can tell you in our mass spec, we didn't see GATA3.
Okay. All right. So another question, I think the question I'm going on is very mechanistic, which is beautiful. I hope this is something you will not make trouble for you with the reviewers, because it seems like a really exciting study. So have you tried abolishing the TOX2 interaction to look at how this affected TH17 differentiation?
No, we haven't. And we really don't. First of all, we don't have enough. Probably we should do more homework to really pinpoint where, like, which residues is responsible for the TOX and TOX2 interaction, right? So we don't exactly know how TOX and TOX2 interact, right? So again, and we don't even know they directly interact or they need something in between. We know they are in the same complex. We know if you overexpress two of them in the cells, you can pull down by co-IP. But again, whether it's direct or indirect, again, there are other protein in the complex, right? So whether it is also through indirectly binding with another protein, or they actually direct leach of that. And of course, more study is needed to identify whether it's direct or indirect, and if it's direct, which residues on TOX and TOX2 is required, and can you just simply disrupt that interaction, as the question was asking? But we haven't come to that part yet.
Fantastic. Have you looked, this is my question, so it's not going to be there. It's just of curiosity. Have you looked at critical enhancers that are regulating TOX2? Do you have an idea of,
Yeah. So first of all, in the TFH study, it has already been shown STAT3 drive TOX2 induction. Okay. So in TFH now, so IL-21 and IL-6 both trigger STAT3, and they can all induce TOX2 induction to some degree. And we also confirmed that in our very first slide. We can induce TFH-like by giving IL-6 or IL-21, and you can get TOX2 induction, but not that high. And if you remember, in our other figure, in our study in the Treg induction part, we also see some level of TOX2 induction if you give TGF-beta in IT rate condition.
Yeah.
So there's some synergy between TGF-beta and IL-1, IL-6 together give you much higher in Ki-17 cells, right? But this is the TGF-beta IL-6 combination. But if you do the other way to drive Ki-17 cells, the IL-1 plus IL-6 plus IL-23, right? The cocktail that considered to promote the pathogenic Ki-17 cell, without TGF-beta, you also drive TOX2 induction. So it seems that it's not just one recipe, okay? But definitely STAT3 is important, is needed. You need STAT3 signal. But then something else like TGF-beta on top of it or IL-1 beta signaling on top of it can further drive the TOX2 induction to the level that we saw in Ki-17 cells.
Wonderful. So again, I want to thank you so much for this beautiful talk and discussing it with the audience and with myself. So we're going to have, as usual, right? You can continue to ask the question through the link in the YouTube. In addition, before everyone leaves us, we still have 80 or so participants. Remember, next week there's going to be another exciting talk by Peter Bors. So please join us on the next week meeting, and please share with all of your colleagues, friends around the world to join us for this wonderful celebration of immunology. And thank you again, LiFan, for this beautiful talk. And definitely not the last time you will join on the Global Immune Talk. Thank you.
Thank you. My pleasure. Thank you. Bye.